Okay, this is part 2 of my lecture on chapter 10, and I did leave off one technique in the sequencing, sizing, and synthesizing part of section 1. So I just wanted to make sure not to forget polymerase chain reaction, or PCR, and it is used in order to amplify DNA or to increase the amount of DNA sample so that it can be studied more easily. And the basic technique involves denaturing, priming, and extending the DNA strand, and it does this repetitively, so then you get a lot of copies of your test DNA that can be analyzed, and every time this cycle of denaturation, priming, extension is repeated, it doubles the number of copies of your DNA for analysis. And it is used in gene mapping, studying genetic defects, as well as cancer, forensics, taxonomy, and the evolutionary studies. It has a lot of applications. Okay, so moving on to section 10.2, we're going to talk about recombinant DNA technology, which has a lot of applications, including medical and industrial. And so recombinant DNA technology involves removing genetic material from one organism and combining it with the genetic material of another organism. And we did this in the lab, in the pGlow lab we did last week. And one of the purposes of recombinant DNA technology is cloning. And so it requires that the donor gene is selected, excised, or cut with restriction endonucleases and isolated. Then that gene is inserted into a vector like a plasmid or a virus that can then insert the DNA into a cloning host, and cloning hosts are usually going to be bacterial cells or yeast, and then the bacterial cell or yeast will then replicate that gene and translate it into a protein product. A lot of times, almost always, the desired product of this technology is a protein. Effective cloning vectors have to have certain characteristics. They need to have the ability to carry a piece of donor DNA. That is called being competent. And they must be readily accepted by the bacterial cell or yeast that is the cloning host. Plasmids are good cloning vectors because they're small, they're well characterized, they're easy to manipulate, and they can be transferred into host cells through the process of transformation. Bacteriophages, also known as viruses that infect bacterial cells, have the innate ability to inject their DNA into bacterial host cells through transduction. So those are why those are common cloning vectors. Okay, so in a cloning vector, you need to have an origin of replication so that it can be replicated in the cloning host. The vector has to be able to accept DNA of the desired size, and the vector typically contains a gene that gives the cloning host or bacterial or yeast cell some type of drug resistance. And that's another thing that we looked at in lab a couple of weeks ago. Some of the medical applications of this recombinant DNA technology include being able to manufacture medications on a very large scale. Those medications include hormones, enzymes, and vaccines. Several examples are insulin for diabetes, human growth hormone, which treats dwarfism, erythropoietin, which treats anemia, factor VIII for hemophilia, and then the HBV. And then there is a table 10.2. It should be purple in your ebook, but it has a whole list of protein products that are made through recombinant DNA technology. Immune treatments, interferons, interleukins, orthoclone, macrophage, tumor necrosis factor, this one's kind of cut off, granulocyte colony stimulating factor, mucogen, and it goes on, then hormones, insulin, TPA or tissue plasminogen activator, hemoglobin A, I already said factor VIII and human growth hormone. Then we have Rh DNase, which is pulmozyme, antitrypsin, PEG SOD, and then lipases. So there are a whole slew of products that are able to be created with recombinant DNA technology. Okay, so this brings us to section 10.3. And in this section, we're talking about genetically modified organisms, also known as GMOs. Genetically modified organisms or recombinant organisms are any organisms that are produced by introducing genes from other organisms or foreign genes into their genome. The most common way that you'll hear them talked about is called GMOs. They're also called transgenic organisms. And so you can have everything from microbes to plants and animals that are GMOs, and you will have recombinant microbes that include bacteria and viruses, and then you can also have transgenic plants and animals. Okay, so when it comes to recombinant microbes, those include bacteria and viruses. There are recombinant bacteria that are used to help remediate environmental disturbances like Pseudomonas syringae, which prevents ice crystals from forming. And then Bacillus thuringiensis is a recombinant bacteria that it has an innate insecticide, and that can also be used. And so all recombinant microbes that are released need to be approved by the EPA or the Environmental Protection Agency, and they are monitored fairly closely. There is a class of recombinant viruses that is being researched to help to cure cancer or to treat cancer. Oncolytic adenoviruses have been genetically altered so that their host range includes cells that produce cancer-specific proteins to lyse and kill the cancerous cells. The USDA or the FDA has not yet approved the procedure for use in humans, but this technology gives hope for the potential of the use of genetically engineered viruses for medical applications. Moving on to transgenic plants or GMO plants. Most of the recombinant techniques used in plants are sort of based on this particular tumor-producing bacteria called Agrobacterium tumefaciens. And these bacteria are just naturally living in the soil, and when plant tissues are injured, these bacteria invade those tissues and they adhere to plant cells and introduce a large plasmid called TI, aka tumor-inducing, and then that TI plasmid inserts itself into the genome of the infected plant cell and transforms it so that it will translate the sequences in that DNA. So even though the TI plasmid naturally is not beneficial, or it's actually harmful to plants, scientists have figured out a way to manipulate that large plasmid and use it as a way to, as a cloning vector for foreign genes that can make plants have herbicide or disease resistance. And so most of this technology has been geared toward really important agricultural crops and giving them herbicide resistance and disease resistance. So the Agrobacterium cells take up the recombinant plasmids or the modified plasmids, and then they multiply and copy the foreign gene. The genetically engineered Agrobacterium is then inoculated or inserted into a culture of target plant cells and infects the cells. Okay, so then that bacterium fuses with the cell wall of the plant. The plasmid enters, the tDNA carries the herbicide gene into the plant nucleus, and then mature plants can be grown from those inoculated cells, and those are transgenic plants that will express the new gene that conveys either herbicide resistance. A lot of them are for herbicide resistance, so you can just spray the whole field and it'll get rid of the weeds, but not the crops. So they'll express that new gene, and the gene becomes part of the plant's genome. So then when the initial crop is grown and produces seeds, then the new gene is then transmitted to the offspring, or it's inherited by the offspring. And so when you grow out those seeds, they have that herbicide resistance or disease resistance. Table 10.3 in your book has a list of GMO or genetically modified plants: cherry tomatoes, wheat, golden rice, soybeans, corn, cotton, peanuts, bananas, plums. And so in cherry tomatoes, they're modified to have high levels of anthocyanin, which is an antioxidant. And what happened was the tomatoes were engineered with a pigment gene from snapdragons that produces 5 times the purple pigment, or that anthocyanin pigment, which increases the nutritional value of cherry tomatoes. Wheat has been genetically engineered to have resistance to head blight, which blight is caused by a fungus called fusarium. A lot of plants are susceptible to fusarium, and so that's made a big difference in wheat production. Also, wheat has been genetically engineered for improved nutrition. Same with golden rice. Soybeans, corn, and cotton have been genetically modified to be resistant to herbicide. Monsanto is one of the companies that's done a lot of genetic engineering, and there's a lot of controversy surrounding it. I'll talk about that in a minute. Peanuts for insect resistance, banana and plum. Banana for vaccine production, and plum for viral resistance. When it comes to genetically modified plants, in 2012, there were about 240 million acres of GM plants that were grown in the U.S., and those were for herbicide and insecticide resistance, mostly corn, soybeans, cotton, and canola. And this has happened in Canada already, but there have been a lot of concerns about the potential negative impacts of genetically modifying these plants. And one of them is just the potential for the genetic recombination to affect wild plants that are nearby, which could create superweeds that are resistant to herbicides and disease. And so in Canada, it's the only place where this has been documented, but canola plants have transferred their genes to wild mustard. And so because of this, the USDA has become more aware of potential problems and is increasing regulations on releases of transgenic plants. Okay, so when it comes to transgenic animals, mice, pigs, sheep, cattle, fish, and poultry have all been genetically modified for agricultural and industrial purposes, as well as for the purposes of researching genetic diseases. The fertilized egg or an embryo in the early stages of development can be transfected by a virus, high voltage, or injection to get the foreign DNA into the genome. And even though the success rate isn't very high, once success is achieved, then the animal will pass that gene on to its offspring. And so transgenic animals will transcribe and translate eukaryotic genes into the desired protein product. Animals have been used as models in order to study human genetic diseases. Mice are a really common study organism. There are mouse models for Alzheimer's, sickle cell anemia, and sheep and goats are used to manufacture proteins and excrete them. There's a technique called CRISPR that is newer, and it has become sort of the most common or chosen method to create mouse models of human disease. And basically it is a tool for editing genes that allows scientists to remove or replace genes pretty easily. There are also a lot of medical applications of this DNA technology, and gene therapy is what describes this collection of techniques where defective genes can be